Welcome to our presentation for BS 2002. In this presentation, we are going to look at a research paper and specifically focus on the effects of glucose and prolactin on gene expression in rat islets. A surge of beta cell replication and the appearance of glucose-stimulated insulin secretion, also known as GSIS, during the perinatal period, which is immediately before and after birth, are essential. Infants who lack beta cell surge are at risk of glucose intolerance and have double the risk of developing diabetes in adulthood. This occurs at the same time as an increase in the level of placental lactogen and prolactin, which stimulates beta cell regulation and GSIS in human and rat islets. It was suggested that beta cell expansion and induction of insulin production by placental lactogen and prolactin ease fetal growth and defend against the development of glucose intolerance and diabetes in the newborn. However, molecular mechanisms by which lactogens increase beta cell mass and GSIS are poorly understood. To test the hypothesis that glucose availability might interfere or adjust effects of prolactin on beta cell mass, interactions of prolactin and glucose were explored in the control of the different expressions shown here. The aims of this presentation are to study the effects of prolactin and levels of glucose on various genes for 24 or 48 hours, and also to discuss how the findings will be helpful to the prevention of glucose intolerance in infants.
Our experiment uses several different methods. One of them, titled The Quantification of mRNA Expression, talks about the process where islets are tested to see how great mRNA is expressed. The process starts off with the islets being washed with serum-free basal media and incubated for either 24 or 48 hours with different concentrations of glucose in the presence or absence of rat prolactin. The islet RNA was then isolated as well as INS1 cell RNA, which was reverse transcribed. The amplicon, which is a piece of DNA formed from natural or artificial amplification events, lengths were averaged at around 60 base pairs. All the primer pairs spanned introns. Negative controls were processed without reverse transcriptase, and all the samples from a single experiment were run using a single polymerase chain reaction mixture. The expression levels were normalized against levels of a protein called actin and were quantified using the comparative threshold cycle method. This table shows the oligonucleotide pairs which encode rat genes. It is the analysis of gene expression in rat islets by quantitative real-time polymerase chain reactions. The rat gene for actin is shown to have the least mean CT threshold cycle islets, while the rat gene TPH1 is shown to have the most at 31 mean threshold cycle islets.
This table represents the effect of prolactin and glucose on gene expression in male rat islets. For prolactin, most of the gene expression decreases in mRNA in the 24 hours, except for prolactin receptor and CDK1. For high glucose, most of the gene expression increases in mRNA in the 24 hours, except for P21, and in others there is no significant effect, for example as for menin and CDK4. For additivity synergy, most of the gene expression has no significant effect except for two of them in the 24 hours, which is prolactin and cyclin D2. For low glucose, there is no increase in mRNA, whereas most of the gene expression increases in mRNA and others has no significant effect, such as cyclin D3, CDK2, and TPH1.
The studies from our report have shown time-dependent changes of islet cell cyclins and cell cycle inhibitors. Linking back to our results from the experiment, during the 24 hours of incubation, the prolactin downregulated the cell cycle inhibitors FOXO1, P27, P57, menin and cyclins D1 and D3, however increasing in CDK1. But it also shows that keeping it for a further 24 hours, making it 48 hours altogether, affects our results. This suggests that time definitely has an impact on the amount of mRNA, as we can see from the results in the example. For high concentrations of glucose, it showed an increase in prolactin receptors P18, cyclin B1, B2, D1, D3, IRS2, IGF2, and TPH1, which were further increased after 48 hours except for cyclin B1, in which it decreased again. In low concentrations of glucose, the genes shown in the table have all decreased amounts of mRNA, which after 48 hours either had no effect or further decreased except in P27 and menin, in which it increased. These findings also suggest that the prolactin induction of cell cyclins came after the downregulation of cell cycle inhibitors.
As we can see from the results, glucose increases islet prolactin receptor expression and increases the effects of prolactin on cell cycle gene expression. The findings can help us in the prevention of glucose intolerance in infants and the development of diabetes between conception and birth. Thank you for your attention. We hope you enjoyed our presentation.